with PL-Pro of MERS coronavirus. So we overexpressed the protein, and once we got the enzyme, because we have been working on with the SARS coronavirus PL-Pro over the years, we had like 4 SARS PL-Pro lead inhibitors.
So you had them in the freezer ready to go?
Yes. So we wanted to see if those SARS PL-Pro inhibitors would work for MERS PL-Pro.
Because while protease sounds like one protease would be the same and one virus would be the same in the other, you don't know whether the inhibitors are going to be active in that other virus.
Right. We don't know until we actually try. So we tested 4 of them, none of which were effective to MERS coronavirus PL-Pro.
None were?
No.
So is this where Dr. Lay comes in? He begins to tinker?
Not yet.
Not yet. Okay.
So, I mean, it was actually parallel because we needed a structure of the APO MERS PL-Pro to see actually how different those SARS PL-Pro and MERS PL-Pro structures are.
So this is really fundamental basic science where you really want to understand how this protease is attacking its substrate. You want to see what's active so that you can then target your inhibitor to the right spot.
Yes. So how started working on the crystallography to solve APO MERS PL-Pro structure, and I started to high throughput screening with our very special compound library.
So was he a very demanding colleague? He wanted you to make lots of this stuff so he could crystallize it, or was that his job?
I think crystallization is mostly my job.
So you had to make the massive amounts and then make the crystal of this molecule and then the inhibitors?
Actually, we're trying to get the co-crystal structure, but at the very beginning, we just want to know like the structure of the protein itself, because we need to know the protein itself first, and then better to know like how it interacts with the inhibitors later.
It's sort of like finding a parking spot in the busy city of Chicago. You got to find where that empty spot is and to target your inhibitor.
Yes. So basically, I character myself more as a structural biologist. Hyun wanted to talk to me like, do you want to like we collaborate and start the MERS PL-Pro project? I was like, yeah, of course, I would like to. So at the very beginning, I did a little bit like cell sequence alignment studies, and I think the MERS PL-Pro and the SARS PL-Pro actually shares a similarity like above 70%, which is fairly high. We just assumed that since the similarity is so high, and I did a little bit of homology modeling, and I believe the structure similarity between them, like the 3-dimensional structure between them should be high too. And also we assume they're gonna have the same inhibitor selectivity, which turns out like is totally ineffective of those inhibitors, of the SARS inhibitors against the MERS. So which kind of surprises us at the very beginning. So later on, after I solved the MERS PL-Pro crystal structure, this even shocks us more. It's because after solving the structure, we made a comparison between the SARS and MERS PL-Pro, and we find the overall structure of these 2 proteins are very similar. And even the catalytic sites, they overlapped very well. So it really doesn't explain at the very beginning how come like those 4 inhibitors against the MERS are not effective at all. So later on, after we thoroughly examine the structure a little bit more, like we're trying to find the answers from the structure-wise, and we find there are 2 amino acid residues from an important loop that are different from the 2 species, which these 2 amino acid residues in the SARS are like forms of chemical interactions with the inhibitors in the SARS, but it replaced by the other 2 different amino acids, which lost the chemical connections with the inhibitors. We believe this is the major reason why those inhibitors are not effective against the MERS.
So Dr. Chan,
yes,
you're on the front line taking care of sick people.
Yes.
And you obviously have seen SARS from the area of the world that you come from.
Yes, that's correct.
SARS emerged out of Asia, and during your clinical career, you probably saw some SARS patients.
Yeah.
And so when you read for the first time about the emergence of MERS, it probably got you very concerned, because this was an agent not endemic to Asia, but endemic to the Middle East, and you were probably faced with, my goodness, I'm going to have to take care of a whole new class of virus for which I don't have drugs. So why don't you share with us how your role in this process came about?
Right. So our team has been working on coronavirus research since the time of SARS. So during the post-SARS era, we have been actively surveying different animal species, especially bats, and we found numerous bat coronaviruses and also other mammalian and avian coronaviruses. And together with the human coronavirus HKU1, which is another human coronavirus, we now know exactly where the MERS coronavirus is situated phylogenetically related to the other coronaviruses. So in fact, at the discovery, we were quite surprised when MERS coronavirus emerged in 2012. We found that the most phylogenetically closely related viruses were the bat viruses, HKU4 and HKU5, which we found in bats in southern China at that time. So we were thinking whether this is again another human coronavirus that emerged from bats.
And that's what I think they're learning out of the Middle East as well, is that there's a bat species that particularly frequents or roosts in tents, if you will, of the nomadic tribes in the Middle East.
Right.
And that may have how it, jumped into the camels and everything as they say.
That is the hypothesis that is being taken now. So the important animal reservoirs that people consider nowadays to be related to MERS coronavirus are the camels, dromedary camels, and possibly bats in the Middle East or in Africa. And to clinician scientists like us, we always want to find a cure for the patients.
You need medicines because that's what people want.
Yes.
Doctor, fix me.
Yes, hopefully. And so in any emerging infectious diseases, time is always against us.
Yeah.
And we can see that the outbreak has grown in the past 2 years. Now it's more than at least 800 cases with nearly 300 deaths. So one of the approach to find new drugs is what the excellent work of Dr. Lee and Dr. Lei have done, is to find specific enzyme inhibitors that would inhibit the replication cycle of the virus. But that would take years or at least months before it can be used in humans.
You need to test it in humans, you need animal studies.
And so the other approach that we could have adopted was to see what is available in hand. And we can do that with two methods. First is we find out what we have been using for treating SARS patients. Although the data is lacking from randomized clinical control trials, we do have some historical case control studies. And some of the examples include interferons and ribavirin, and also lopinavir, which is actually another protease inhibitor that you have mentioned in the treatment of HIV-infected patients. So we tested these drugs, and we found a number of interferon preparations, and we found that betaferon among them is the most potent in inhibiting MERS coronavirus. And to give you a perspective of how potent it is, the IC50, which is the 50% of the concentration of the drug that is needed, the concentration of the drug that needs to inhibit 50% of the virus infection in vitro, is around 17 international units per mil. Now, what could be achieved clinically as the peak serum concentration after a subcutaneous dosage is about at least 2 times higher than this. So that makes it a possibility. And actually, betaferon or interferon beta 1b has been frequently used in patients with multiple sclerosis. So we would know quite well the PK/PD profiles and also side effect profiles.
And safety.
Yes,
you
exactly.
know whether or not it would be safe to
and
use.
the dosing regimen and so on. And then we also perform another program called drug repurposing program. And basically what we do is we use an automated robotic machine to screen a chemical library consisting of more than 1,000 drugs that is available commercially and is also FDA approved for other purposes, to see whether they also have anti-MERS coronavirus activities. And what we found is that mycophenolic acid, which is available usually as mycophenolic mofetil or mycophenolic sodium salt, and is used usually as an anti-rejection therapy in patients with organ transplantation, was highly effective. And when we compared mycophenolic acid with ribavirin, because ribavirin was another drug that was used in SARS, and also it's a very broad spectrum antiviral, we found that the IC50 to Cmax ratio is much better in the mycophenolic acid. So theoretically, if you calculate everything, you make the calculations, we show that mycophenolic acid only after a dose of 1 gram orally, which is not a very high dose, and it's clinically relevant, the concentration in the serum should theoretically last for at least 5 days higher than the IC50 for MERS. Now, but there's one particular concern about the use of mycophenolic acid in such a case, because mycophenolic acid has some immunosuppressive effects, right? Obviously,
I was going to ask you about that.
We don't want to.
Sort of the double-edged sword.
So, further animal studies should be confirmed to address this issue. But we believe if you can only give 1 small dose at the beginning of the MERS infection to the patient, and then you just stop it and you wait, because the dosage that gives the peak serum concentration to be above the IC50 should last for about 5 days already. And that should not have a very significant side effect as in organ transplant recipients who are regularly taking the drug. But again, everything I've said are postulations, so we have to confirm in further animal studies and hopefully clinical trials.
I suppose that's the good news, bad news piece for your story is SARS was blessedly short-lived as an epidemic, and then as a virus that we are concerned with, it seemed to burn out fairly quickly. But what your studies are showing is that work from Dr. Lee's group is very important.
It's very important.
And developing these new protease classes, because there are many more bats out there with many more coronaviruses, and perhaps we are going to set up a collaboration here from the couch where you can actually begin to test some of the compounds in her library to see whether or not they're active against some of these other coronaviruses. So as an emerging graduate student, what do you think of all this stuff about drug development and looking at crystallography and all of these things?
Nowadays, crystallography actually is a well-established method widely used in the pharmaceutical industry, in the academia. So I don't know if everybody is familiar with that or not. Basically, it's very easy. It's like you use microscope to observe the bacteria, but now we wanted to observe how it looks of the protein structure. But there actually is no such microscope can enlarge until you can see the structure of the protein. So we use X-ray to diffract the protein, and we collect the data, and we reconstruct the three-dimensional image by the computer programs. So this is a very visually direct, can visualize like how the protein looks like, and if you're lucky, you get the co-crystal structure with your inhibitor with the protein. You can even get the information like how the inhibitors interact with the proteins. I think in the drug, not in the drug stage, but in the inhibitor optimization stages, like you want to expand the chemical scaffold, you want to build more interactions, enhance its binding affinity or inhibitory affinity. This relies a lot on the crystallography studies, because you can, based on the interaction patterns, you can